Hi, welcome to the recording of Lecture 9, Skeletal Muscle. My name's Kelly Linden and I usually take the Aubrey internal lectures, but unfortunately I was unwell and missed one lecture this week, so I'm just recording this one from my office. Now, this lecture on skeletal muscle, there'll be a fair bit of overlap from any first year physiology subject that you would have done in your courses. So most students in BMS256 did BMS161, and so if any of this material isn't making sense, you might like to have a read over the chapter of your first year physiology textbook, like Sealy, whichever textbook you used, just to sort of refresh your memory of what happens with skeletal muscle. This information is also available in chapter 18 of the prescribed text McArdle Catch and Catch. Now, in lecture 10, which is control of skeletal muscle, that will be chapter 19 of McArdle Catch and Catch, and we really have to have a good understanding of this lecture of the structure of skeletal muscle and the basic physiology before we can understand the control well. Now, as I said, I'm recording this lecture in my office and I've been a bit unwell this week and it's Friday afternoon, so I'm probably not setting myself up to make the most exciting recording ever. So I have a picture on the other screen of the computer of my two dogs, and I'm pretending I'm presenting it to them. They always find what I'm saying interesting, so hopefully I'll be able to interest you in the topic also. Okay, now as per usual, we've got the objectives listed here, and the exam questions will come from the lecture objectives. So, and there's only 4 for this slide set. So we'll list the levels of organisation of skeletal muscle, and that will be a lot of revision from BMS 161. We'll also describe the blood supply to skeletal muscle and how it can change with endurance training. Now, I think I need to pause here a little bit, not literally pause, but just digress and mention that with this subject so far, there's been quite an emphasis on changes that we might see with endurance training, and I'll sort of talk about with the Olympics coming up and refer a lot to this with elite sport. It's really, really important to bear in mind that all of the changes that we see, or most of the changes that we've been talking about, okay, they might make an elite sports person good enough to become an Olympian, or an Olympian win a good gold medal, but they might also help your grandma, say if you had a grandma with COPD who couldn't really was borderline for living at home by themselves or going into care. With some exercise training, all of these adaptations can make the tasks of daily living much easier also. So we see these changes in most people in response to endurance training. So it's not only talking about this in reference to athletes. From weeks 1 to 6 of this subject, we're mostly talking about sort of young, healthy people. But in the last 3 weeks of the subject, from weeks 12 to 14, we'll be talking about all of these changes and how they can help people in special populations improve their quality of life. Anyway, sorry, back to the objectives. After we describe the blood supply to the skeletal muscle, we'll talk about the sequence of events of excitation, contraction, coupling. Again, that'll be quite a lot of revision from BMS 161, but we have to have a really good understanding of that to understand this topic and also later topics in BMS 256. And then we'll contrast slow to fast twitch muscle fibers, and we'll also consider some of the subdivisions. Excellent. Now, we'll just start by talking about the gross structure of skeletal muscle. Now, each of the body's skeletal muscles contain various layers of wrappings of fibrous connective tissue. And remember that each muscle is made up of thousands of cylindrical cells called muscle fibers that lie parallel to each other, and the force of action is directed along the fiber's long axis. Now, during development, these were individual immature cells called myocells, and they've fused to form myofibers, or skeletal muscle fibers. So they've multinucleated. They've many nuclei that lie along the length of the muscle membrane. Now, the fiber length of any given skeletal muscle fiber can vary from a few millimeters, such as the extrinsic muscles of the eye, to nearly 30 centimeters in the large muscles of the leg. And sartorius is the longest muscle and it's located in the thigh. And for a tall person, that can be quite a bit longer than 30 centimeters even. Okay, so we'll just talk about the levels of organization in skeletal muscle. So these are the layers of connective tissue wrapping of a muscle. Now, a whole muscle is surrounded by the epimyosin, and this can be called the deep fascia, and this will separate. Pardon me, and this epimyosin will blend into the intramuscular tissue sheaths to form the tendons. So that surrounds a whole muscle. You can see that just here. Now, within a whole muscle, the muscle fibers are separated into bundles of tens to hundreds of muscle fibers that we call a fascicle, and each fascicle is surrounded by a layer of connective tissue called the perimyosin. Now, within each fascicle, we have, as I said, tens to hundreds of muscle fibers, and each individual muscle fiber is then surrounded by a layer of connective tissue called the endomyosin. So I like to think of it that the epimyosin keeps the whole muscle together, surrounds the whole muscle. The perimyosin is really important as a layer of scaffolding because we're going to have so many capillaries and nerves in the whole muscle. We need these layers of connective tissue for the of scaffolding pretty much for the vascularization and also the nerve supply. Then each individual muscle fiber, as I said, is surrounded by the endomyosin, and I like to think about this as electrical insulation because remember, within a skeletal muscle we have voluntary control over which muscle fibers we contract. So if we wanted to contract one given muscle fiber but not the one next to it, that means that they need to be all surrounded and electrically insulated from one another so the action potential doesn't jump from fiber to fiber. And so the endomyosin performs that function and also, again, allows scaffolding. So an axon terminal and capillaries can run through next to every muscle fiber. Oops, just missed a little bit of animation there. Cool. Okay, now if we go through the levels the levels of organization of skeletal muscle to one level deeper, we'll get the muscle fiber membrane. So the plasma membrane of a muscle fiber is called the sarcolemma. Now the sarcolemma surrounds each muscle fiber and encloses the fiber's cellular contents, just like the plasma membrane does for cells. For what we could call our typical cell that we would have learnt about in cells lectures in BMS256. Okay, the cytoplasm of muscle fiber is called a sarcoplasm, and this contains the nuclei and also the nuclei also contains the genes, obviously. Then the sarcoplasm, within the sarcoplasm, will also have many mitochondria because it's a cell that produces a lot of ATP and other specialized organelles. Now, the last thing that we need to make sure that we're familiar with is the sarcoplasmic reticulum. Now, this is a modified endoplasmic reticulum, and it stores calcium within a muscle fiber and also provides some structural integrity to the cell. Okay, so we have the picture here. We can see a muscle fiber. Point out all of the little bits of that in a moment. Okay, now blood supply to skeletal muscle. We mentioned in the cardiovascular lecture that in lectures 7 and 8, also in week 3, that because skeletal muscle fibers have a lot of mitochondria and they produce a lot of ATP in the mitochondria through oxidative phosphorylation, they need to have a good blood supply so that a lot of oxygen and other nutrients can be delivered, and we need efficient removal of carbon dioxide and other waste products from the muscle fiber. Otherwise, it's not going to be able to generate ATP at the rate that we need, and we won't be able to contract the muscle the way that we want to. So we have a high blood supply to our skeletal muscles. Now, within a muscle, arteries and veins lie parallel to the individual muscle fibers, and they divide into numerous arterioles, capillaries, and venules to form a network around the endomyosin. Because each endomyosin, so the connective tissue layer that surrounds an individual muscle fiber, needs to have capillaries nearby so that we can have effective transfer of oxygen and nutrients into that muscle fiber, and again, the removal of wastes. Now, during intense exercise, the vascular bed delivers large quantities of blood through active tissues to accommodate this increased oxygen need. So, again, we spoke about this in the cardiovascular lecture. The oxygen, we have an vasodilation of the blood vessels going to active muscles, while we shunt blood away from areas where we don't need to send it, such as the digestive system and kidneys. During exercise. Now, there's a difference in the blood pressure response during exercise and physical activities that require straining. So, especially some large static movement, say if you're holding a squat. Involves a number of large muscle groups, and as you're contracting the muscle, it actually occludes the local blood flow to that muscle. So if you hold that contraction for a long time, you can really spike your blood pressure up quite high. There's less of an effect for straining contractions for smaller muscle groups. So, say in my instance, my bicep is not large, we could say. So if I was straining and holding a bicep curl, then my blood pressure spike would be quite a bit less because I'm occluding a smaller area. Then with dynamic exercises like running, cycling, rowing, where we've got contract, relax, the blood pressure spike is less again. So these are really important considerations for people when prescribing exercise for people with hypertension. Say we want to avoid those really big blood pressure spikes, but we want to encourage some small changes. So dynamic exercise is often a bit safer. Okay. We can consider capillarization at skeletal muscle, and we see with endurance training, we see an increased number of capillary to muscle fiber ratios. So after approximately 2 weeks of exercise training, you actually release a growth factor called VEGF, vascular endothelial growth factor, and that starts to increase angiogenesis, so the number of capillaries at any muscle bed that you're training. So if you're just cycling, then you're not going to have this increasing in your arm, or say in your biceps. It will be more specific to muscles that are actually contracting during cycling, such as vastus lateralis. Pardon me. Now, this is one of those things that as soon as you stop endurance training, it reverts back to normal. All right, so this enhanced capillary microcirculation expedites the removal of heat and metabolic products from active tissue, and also facilitates delivery of oxygen, nutrients, and hormones. So it's one of those adaptations. So when you start exercise training, you might, I'm sure everyone's been familiar with this at some stage. If you start doing an exercise that you haven't done before, using muscles that you haven't really used before, you often feel the burn before your heart, like you feel like you're really huffing and puffing. So it feels like your muscles fatiguing quicker than the rest of your body is, like your cardiovascular system or respiratory system, and that's often because you don't have so many capillaries in that region. But after endurance training, so even after a couple of weeks, that feeling should start to subside somewhat because you've got increased capillaries already in that area, so we can deliver more oxygen more quickly during exercise. The total number of capillaries per muscle averages 40% higher in endurance trained athletes than untrained counterparts. So we end up with a lot more capillaries with endurance training. Now, the things that cause this are vascular stretch and shear stress on the vessel walls from increased blood flow during exercise stimulates capillary development with intense aerobic training. And again, that's because of this growth factor, VEGF, vascular endothelial growth factor. Okay, now this next slide isn't skeletal muscle ultrastructure, or we could call that microstructure also. So a single multinucleated fiber contains myofibrils that lay parallel to the fiber's long axis. So if we look at this diagrammatic image here of a skeletal muscle fiber, we can see it's multinucleated. So this is only a section of a skeletal muscle fiber, and each of them have two nuclei in there, and we can imagine if this muscle fiber was longer, we'd see many more nuclei. Now the nuclei are located to the periphery. They're kind of pushed to the periphery because inside the middle part of your muscle fiber contains the contractile apparatus, the proteins that we need to generate force in the muscle. So that's just pushed the nuclei to the edge. And we see that the muscle fiber is striated, so it's got alternating regions. Okay, so we'll just talk a little bit more about actin and myosin. Now, thousands of myosin filaments lie along the line of actin filaments in a muscle fiber. So the myosin filaments are what we call the thick filaments, and we see they look kind of like, somewhat like a golf club. Um, so they consist of bundles of molecules with polypeptide tail and the globular head. So they're part of the golf club that would hit the ball. Uh, Okay, so if we look at the structure of a sarcomere, so a sarcomere is the functional unit of a muscle fiber, and we can see, and this is what gives that striated appearance, the alternating dark and light bands in a muscle. So the light bands are going to be the thin filament, the actin, which is shown here in blue, and the the dark bands will be the thick filament, the myosin, we can see here. Now, a sarcomere is one Z line, which is the structural protein, made of alpha actin that secures actin in place. So it's one Z line to the next Z line. So that's one sarcomere, and that's the functional unit of a skeletal muscle. In physiology, we consider the functional unit as being the smallest region or smallest area in that organ that can provide. Okay, now cross bridges. Um, so now we're going to consider how actin and myosin contract together to form cross bridges. So the globular myosin heads extend perpendicularly to latch onto the double twisted actin strands to create structural and functional links between myofilaments. So during a muscle contraction, when the regulatory proteins have been moved off actin, the myosin head, the globular head, is going to bind to the actin molecule. So ATP hydrolysis activates the myosin's two heads, placing them in an optimal orientation to bind actin's active site. So we need ATP to split the myosin head, ATP to split to move the myosin head, and this then pulls the thin filament to the Z lines towards the center of the sarcomere, so it shortens the sarcomere. Now, tropomyosin and troponin regulate the make and break contacts between the myofilaments during muscle contraction. So we can see in this little animation at the top here that ATP is binding to myosin. It then moves its head, binds to actin, and then relaxes again. So again, ATP binds to myosin, it binds to actin, and it moves. And this is all acting to... Oh. Okay, with the sliding filament model for muscle contractions, the thick and thin filaments slide past one another without changing length. This produces a change in relative size of the muscle without the sarcomeres shortening. Without the actin and the myosin actually shortening, but the whole sarcomere shortens. And if every sarcomere along the length of a muscle fiber shortens, we see a shortening of the whole muscle. Okay, we'll just play this short video that explains the interaction of actin and myosin.
Filaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Excellent. And there's plenty of other videos like similar to this on YouTube. If you would like to see more animations. Okay, and here's just a figure from your textbook showing cross bridges formed from the myosin filament binding to exposed actin. Okay, so we're now just going to talk through the sequence of events that we see during a muscle action. Okay, so for the first step, we see generation of an action potential in the motor neuron. This causes the terminal axon to release acetylcholine. So remember that every skeletal muscle fiber must be innervated by a motor neuron, by an axon terminal of a motor neuron. So the action potential will travel down there, release acetylcholine at the neuromuscular junction, and this will bind to specialized acetylcholine receptors on the sarcolemma. Um, so for step 2, the muscle action potential depolarizes the transverse tubules. Um, and we can work ahead to step 3 here. So we can see this action potential is going to travel down the length of the sarcolemma and then down the T tubules. And once the T tubules are depolarized, this causes calcium to be released from the lateral sacs of the sarcoplasmic reticulum. Now, when we see the the T tubule and a sarcoplasmic reticulum on each side with calcium stores, I've got in pink here, we call this a triad. Okay, and so if I animate this slide, the action potential will travel down the T tubule, and this will depolarize the T tubules and cause calcium to be released into the sarcolemma. Now, when calcium has been released, it can bind to the regulatory protein troponin and allow actin and myosin to bind to form cross bridges and generate force in the muscle, as indicated by this little doobie whacker here. Okay, so when calcium has been released, it binds to the troponin and tropomyosin complex in the actin filaments, and remember that complex was preventing actin from binding with myosin. So this releases the inhibition that prevented actin from combining with myosin. So now actin and myosin are able to bind to one another. So for step 5, actin combines with the myosin and also an ATP molecule. And this also activates the myosin ATPase, which is the enzyme that splits the ATP in the myosin head. This reaction's energy produces myosin cross-bridge movement and creates tension. Now, when ATP binds to the myosin cross-bridge of actin and myosin, this breaks the actin-myosin bond and allows the cross-bridge to dissociate. So it releases actin. The thick and thin filaments then slide past each other and the muscle shortens. Now, for step 7, the cross-bridge activation continues when calcium concentration remains high enough to inhibit the troponin tropomyosin system. So as long as action potentials continue to travel down the length of the muscle fiber, then calcium will be continually released from the sarcoplasmic reticulum, and this process will continue. But the muscle relaxes as soon as the skeletal muscle action potential ends and the calcium is immediately taken back up into the sarcoplasmic reticulum. Now, when the calcium is removed back into the sarcoplasmic reticulum, this restores the inhibitory actin of troponin and tropomyosin. They return to the actin molecule and prevent it from interacting with myosin. Now, in the presence of ATP, actin and myosin remain in the dissociated, relaxed state. Okay, now the last thing that we're going to talk about today, or the last topic, is muscle fiber type. So there are two main types skeletal muscle contains two main types of fibers that differ in the primary mechanism that they use to produce ATP. the type of motor neuron innervation that they have, and the type of myosin heavy chain that we have expressed in them. So within the myosin molecule, there's different isoforms, so different variations in the heavy chain, which is part of the molecule, and that really affects the function of how the myosin work. Now, the proportions of each type of muscle fiber vary from muscle to muscle and from person to person. Now, what we know about muscle fiber type usually comes from muscle biopsies, and I've been mentioning this series of photos in lectures. It's a shame I can't present this in front of the whole class, but here we have a subject who is participating in an exercise trial. So this is Professor Glenn McConnell over here. Doesn't necessarily look like the lab head, but he is. This is a professor of medicine at Monash Uni, Ben Canny. He's about to take a muscle biopsy. So, and this is a research participant. So he's been cycling for 2 hours, and we need to take a muscle biopsy within 7 seconds of him stopping exercise. So we can see he's got tubie grip on his thigh here, and we can see a little bit of a bandage here because Ben Canny, the medical doctor, will have already made an incision for each muscle biopsy with their scalpel blade through the fascia of the muscle. And so as soon as the subject stops exercising, he lies straight back on the bed, and Ben pulls off the tubie grip and exposes the incision that he's already made and puts this biopsy needle, which is kind of the thickness and bluntness of your index finger. Now the biopsy needle has a little knife on the end, and then this apparatus here is some suction, so the muscle moves into the needle through a little window and then is sucked up up away from the window, so that the knife action can continuously cut muscle. Don't take very much, it's only like a few grains of rice, but we can see here the needle's now in the subject's thigh. Okay, and that's how we take a muscle biopsy. So it's a little bit of a gruesome procedure. And then there's experiments that you can do to test for what muscle fiber type, what proportion of each muscle fiber type somebody has. Okay, now so that's how we know a lot of what we do know from about muscles from human muscle biopsies, but we can also study muscles in these sort of organ bath style setups here. And in this study, I used to dissect out the EDL and the soleus of mice. Now the EDLs are a very red, very slow twitch muscle, and the soleus is a very slow twitch muscle, and the EDL is a very fast twitch muscle in a mouse. So we can tie a suture to each tendon of the muscle and fix one to a fixed pin and the other to a force transducer, and we can leave these muscles in these little organ baths that have all these tubes coming in and out, so they're supplying oxygen to the organ baths, and we can contract the muscle, and we can learn a lot about muscles and different muscle fiber types from doing these sorts of experiments. Okay, here we have a table that classifies different human skeletal muscle fiber types. Now, it classifies these into type 1, type 2A, type 2X, and also type 2B fibers. So we've got four different types of muscle fibers that we need to learn about. Now we can think about type 1 as being slow twitch, that's the easy one, but then there's sort of 3 levels of fast twitch fibers. So we go from moderately fast to very fast as we move from type 2A to 2X to 2B. Now, in this next column, size of motor neuron, the size of a motor neuron, the diameter the size of the motor neuron is going to influence how many, the size of a motor neuron is going to influence how many muscle fibers are innervated. So with our type 1 fiber, we've got a much smaller number, and as the muscle fiber gets faster and faster, we have more and more. Now, with our slow twitch fibers, we have a high resistance to fatigue, so we can call these muscle fibers fatigue resistant. Whereas with all of the fast twitch fibers, they fatigue more quickly, so they can't undergo continuous contractions like a fast twitch, like a slow twitch fiber can. But we've got as we move from the slowest of the fast fibers, we have some fairly high resistance to fatigue to intermediate to low. Now, as for the activity used for the different fiber types, for type 1, our slow twitch fibers, they're aerobic fibers, and the maximum duration of use can be for hours, but the force production is much lower than of the other varieties. Whereas our type 2A fibers are long-term anaerobic, so they can be used for activities that last less than 30 minutes, and the force production is medium. Whereas our short-term anaerobic fibers can only be used for activities less than 5 minutes. The force production is high, and then short-term anaerobic, they fatigue very, very quickly, so they can only be contracting maximally for approximately 1 minute, but the force they produce is very, very high. So if you consider, say, with the Olympics coming up, let's think about some Olympic sports. Let's pick athletics. So if you had a higher proportion of type 2B fibers in your legs as a sprinter, then you'll be able to generate more force than someone that has a higher proportion of type 1 fibers, because they'll be able to continue for hours. But it's important to also, so if we think about sports, say 100, 200 and 400 meters at the Olympics on the flat, for athletics, are all less than 1 minute. Whereas type 2X fibers, we can start to consider longer running races like the 1500 meters. Type 2A, we can consider 5 and 10 kilometer events, and then the type 1 fibers would be beneficial for hours, so say with the marathon. Now the speeds get a little bit lower as you move from each of these sports, so the average speed for say a 100-meter sprint in the men's race at the Olympics will be about 36 kilometers an hour, so that force production is very high. But then if we move across to say that the marathon's going to be run in just over 2 hours, and it's 42 kilometres. So the athletes are running approximately 20 kilometres an hour. So even though here we say the force production is low, we consider it lower than type 2 fibers. It's not the fact, if you had a high proportion of type 1 fibers, it doesn't mean you're really, really slow. It just means you probably can't sprint really, really fast. You can still run quite fast. 20 kilometres an hour is still very fast to run. Now, with the rest of this table, it's referring to this main the main substrates and generation of ATP for these muscles. So if we consider type 1 fibers, they're highly adapted for oxidative phosphorylation. So we have a high density of mitochondria, we have a high capillary density because we need lots and lots of oxygen to deliver, for our mitochondria to generate ATP. Whereas there's low glycolytic capacity because most of the ATP will be produced through the citric acid, through oxidative phosphorylation in the mitochondria, the citric acid cycle and electron transport chain. And the major storage for fuel in these type 1 fibers is triacylglycerol, or triglycerides, parts of our fatty acids. A glycerol plus fatty acid. Now this is going to be mostly used again in the citric acid cycle and in the electron transport chain to generate ATP. Now if we go to the other end and go to the fastest of the fast twitch fibers, the type 2B, we'll see that the mitochondrial density is low because they're not going to use very much oxidative phosphorylation, And likewise, the capillary density is low because they won't need all that much oxygen, and oxidative capacity is low, whereas glycolytic capacity is high, and they'll have a whole lot of creatine phosphate and glycogen stores, so they'll be great for the first minute or so of exercise where we're using phosphocreatine and glycogen through glycolysis. Now, with our intermediate fibers, the 2A and 2X, they've got intermediate amounts of these substrates because they're somewhat in between. Now, with myosin heavy chain, these are just human genes, and it's just indicating that there's a different gene for each type of muscle fiber. And so the type of muscle fiber that we have is largely determined by the gene, the type of and the type of myosin that we're expressing in that muscle fiber. Okay, so we'll just summarize each of these muscle fiber types. So fast twitch fibers we can call type 2 collectively. So they were 2A, 2X, and 2B. They exhibit the following four characteristics. They have a high capacity for electrochemical transmission of action potentials. So the action potentials are traveling at a high rate along the sarcolemma. They have a high myosin ATPase activity. If we consider that they're generating more force, that means they need more actin and myosin cross bridges, and these are occurring at a very, very fast rate. So actin and myosin are binding together, then releasing and binding and releasing at a much faster rate than in a slow twitch muscle fiber. And we also have a rapid release of calcium and from the sarcoplasmic reticulum, and also a very efficient uptake in between muscle contractions. Now, these factors contribute to this fiber's rapid energy generation for quick, powerful muscle actions. Okay, now the fast twitch fiber's intrinsic speed of shortening and tension development ranges 3 to 5 times faster than a slow twitch fiber. So they really rely on a well-developed short-term glycolytic system for energy transfer, for generating ATP. So fast twitch fiber activation predominates in anaerobic type sprint activities and other forceful muscle actions that rely almost entirely on anaerobic energy metabolism. Now, activation of fast twitch fibers plays an important role in the stop and go or change of pace sports such as basketball, soccer, lacrosse, field hockey, football, netball. Okay, and just as a summary here, we have the three types of type 2 fibers, and these are the primary subtypes. Sorry, so we have type 2A represent the fast oxidative glycolytic fibers. So these are the most intermediate of the fibers. They're very similar in many ways to a slow twitch fiber, that they just fatigue a bit more quickly and don't quite have the oxidative capacity. But these can train really well. So if you do a lot of endurance training, they can increase in the number of capillaries and increase their oxidative capacity. So these can become quite type 1 like. Type 2X possess the greatest anaerobic potential and most rapid shortening velocity, and they're the true fast or glycolytic fiber. And then we also have type B fibers. We'll keep moving on. Okay, so slow twitch fibers, or type 1, generate energy for ATP resynthesis predominantly through the aerobic system of energy transfer. And their four distinguishing characteristics are they've got low myosin ATPase activity, so the myosin breaks down actin at a much slower rate, and this is why they generate less force. So myosin will split ATP, then it will bind to actin, and then it will release, and this all occurs much, much more slowly than in a fast twitch fiber. They have a slower calcium handling ability, so when an action potential travels down the T-tubules, calcium is released from the sarcoplasmic reticulum more slowly, and then it will be pumped back into the sarcoplasmic reticulum more slowly also. They have a less well-developed glycolytic capacity than fast twitch fibers, so naturally they just have less phosphocreatine and enzymes involved in glycolysis. But with serious sprint training over time, the type 1 fibers can become more fast-like, so you can increase their phosphocreatine stores and increase the number of glycolytic enzymes in them, so they have more anaerobic capacity, but they'll never be like a true fast twitch fiber. Now, type 1 then have also large and numerous mitochondria for really high oxidative capacity. So they're highly fatigue resistant and ideally suited for prolonged aerobic exercise. Um, so they're slow oxidative fibers with slow shortening speed and rely on oxidative metabolism. And both slow and fast muscle fiber types contribute during near maximal aerobic and anaerobic exercise. So that's important to remember, that we don't just contract type 1 when we're going for a long jog and type 2 when we're at the gym or doing some explosive exercise. Near maximal exercise, where for aerobic and anaerobic, we're contracting all of them. Okay, so this is just a figure so from, if you were to take muscle biopsies, you can stain for different fiber type, and it's literally just a matter of staining for the different fiber types and then counting through how many slow twitch fibers somebody has to how many fast twitch they have. And the fast twitch, it depends on the stain actually. So I won't talk about that too much, get into the science, but there is a description in your textbook of the staining science. It's quite an easy stain to do. I think there's a few medical science distance students that might find that section of your textbook interesting. Okay, just the final part of this lecture is fiber type differences. So men, women and children on average possess between 45 and 55% slow twitch fibers in their arm and leg muscle. So you can think on average we've got about 50-50 for slow and fast twitch fibers. Now, in athletic groups, that's where we can start to see clusters of different muscle fiber types. So certain patterns of muscle fiber distribution appear in comparison among highly proficient athletes. So, for example, endurance athletes usually possess predominantly slow twitch fibers, whereas fast twitch fibers predominate for elite sprint athletes. Now, performance success is dependent not only on muscle fiber composition, but on a blending of many physiological, biochemical, neurological, and biomechanical support systems. So, just because you've got the right muscle fibers doesn't mean that you'll be great at that sport, but in some instances it can help a lot. Pardon me. Now, larger muscle fibers in male athletes and a larger total muscle mass are a principal gender difference in muscle morphology, and we'll talk about that in our gender differences in exercise lecture in week 12. Okay, so finally, we've just got a slide here that shows the the correlation of muscle fiber composition, the percent of slow twitch fibers in some different sporting groups, and correlating this to their maximal oxygen consumption. Now, traditionally, we say the group with the highest VO2 max is cross-country skiing, because you're using almost every muscle in your body, and it's a really hard endurance training. So we'll see in elite cross-country skiers, they've got the highest VO2 max of any group, so it's on average in the 80s, and they've almost 80% slow twitch muscle fibers. So if you wanted to be an elite cross-country skier, you really need a high proportion of slow twitch fibers. Long distance running comes in second, so they've got, as a group, their VO2 max is very similar, so it's almost 80%, and they've got, say, around about 65% slow twitch fibers. Canoeing is another high VO2 max, so around about 70, and 60% slow twitch fibers. Then we can skip down to non-trained athletes, and we can see the error bars here. So this is indicating that we're between 35 and 80% for the non-trained athletes, and we're going to see VO2 maxes. This seems a little bit, sorry, trained non-athletes of between 50 and 65 and 70. That's a very high range there. I'm not sure what population that's from. And then as we move down to swimming, training, weightlifting, alpine skiing, wrestling, ice hockey, that we're seeing a reduced number of slow twitch fibers and on the whole a reduced VO2 max. And then for running, 100 to 200 meters, so athletic sprinters, we're going to see a less than 50% on average proportion of slow twitch fibers, so more fast twitch, and again a lower VO2 max. Awesome. Thanks for listening. I'm sorry that I had to cancel the lecture this week. Anyway, all right, thanks. Bye bye.